Hello everyone, welcome to our channel. I am Srijita Ghosh. I am your tutor for today's lecture. Food microbiology. We'll discuss this particular topics which includes microorganisms in food, food preservation, foodborne illness, fermented foods. Microorganisms in food. Factors affecting microbial growth in food. Water availability. In general, lower water activity always inhibits the microbial growth. Water activity can be lowered by the process of drying and by the addition of salt or sugar. There are osmophilic microorganisms which prefer high osmotic pressure and xerophilic microorganisms which prefer low water activity. Physical structure. Grinding and mixing increases the surface area and distributes the microbes, which promotes microbial growth. Outer skin, if peeled out of vegetables and fruits, generally slows down the microbial growth. Antimicrobial substances. Coumarins. These are a group of natural compounds and have been found to have multi-biological activities such as anti-HIV, anti-tumor, anti-hypertension. These are mainly found in fruits and vegetables. Lysozyme. Found in cow's milk and eggs. It is an enzyme that digests and breaks down the cell walls of bacteria to improve protein or nucleic acid extraction efficiency during the cell lysis. Aldehydic and phenolic compounds are found in herbs and spices. Allicin, which is generally found in garlic. It is an organosulfur compound obtained from garlic, which is a species in the family Alicaceae. It is the key ingredient responsible for broad spectrum of antibacterial activity in garlic. Polyphenols are found in green and black teas. Food spoilage, which result from the growth of microbes in food, that alters the food visibly and in other ways, rendering it unsuitable for consumption. It involves predictable succession of microbes. Different foods undergo different types of spoilage processes. Even toxins are sometimes produced. Algal toxins may contaminate shellfish and finfish. Composition and pH. Putrefaction. It is the decomposition of organic matter, especially the typically anaerobic splitting of proteins, proteolysis, by bacteria and fungi, with the formation of foul-smelling amine compounds. pH impacts makeup of microbial community and therefore types of chemical reactions that occur when microbes grow in food. Toxins. Ergotism. This is a toxic condition caused by the growth of a fungus in grains. Aflatoxins. Carcinogens produced in fungus-infected grains and nut products. Fumonisins. Carcinogens which are produced in fungus-infected corn. Removal of microorganisms. Usually achieved by filtration, commonly used for water, beer, wine, juices, soft drinks and other liquids. The next topic is food preservation. Low temperature. Refrigeration at 5 degrees Celsius retards but does not stop microbial growth. Microorganisms can still cause spoilage with extended spoilage. Growth at temperatures below -10 degrees Celsius has also been observed. Next process is canning. Food heated in special containers which are known as retorts to 115 degrees Celsius for 25 to 100 minutes. It kills spoilage microbes but not necessarily all microbes in food. Spoilage of canned foods. Spoilage prior to canning, under processing, leakage of contaminated water into cans during cooling process. Pasteurization. It is the process of heating a food, usually a liquid, to a specific temperature for a definite length of time and then cooling it immediately. It kills pathogens and substantially reduces number of spoilage organisms. Different pasteurization procedures heat for different length of time. Shorter heating times result in improved flavors. Reduced water activity. By drying, freeze drying, which is also known as lyophilization, addition of high concentrations of solutes such as sugar or salt. Chemical based preservation. GRAS, GRAS, chemically agents generally recognized as safe. Radiation. Ultraviolet UV radiation used for surfaces of food handling equipment does not penetrate the foods. Radaperdization. Uses of ionizing radiation, gamma radiation to sterilize the meat, seafoods, fruits and vegetables. Kills microbes in moist foods by producing peroxides from water. Peroxides oxidize cellular constituents. Microbial product based inhibition. Bacteriocins. Bactericidal proteins active against related species. Some form pores in plasma membrane. Some inhibit protein or RNA synthesis. Example Nisin, used in low acid foods to inactivate Clostridium botulinum during canning process. Foodborne infection. Ingestion of microbes followed by growth, tissue invasion or release of toxins. Foodborne intoxications. Ingestion of toxins in foods in which microbes have grown. Include staphylococcal food poisoning, botulism, serious paralytic illness caused by botulinum toxin, which is metabolic waste produced under anaerobic conditions by the bacterium Clostridium botulinum and affecting a wide range of mammals, birds and fish. Clostridium perfringens food poisoning and Bacillus cereus food poisoning. Detection of foodborne pathogens. Culture techniques. Immunological techniques which are very sensitive and molecular techniques. Probes used to detect specific DNA or RNA and sensitive and specific they are. Detection of foodborne pathogens by PulseNet, which is established by Centers for Disease Control, uses pulsed field gel electrophoresis under carefully controlled and duplicated conditions to determine distinctive DNA pattern of each bacterial pathogen. It enables public health officials to link pathogens associated with disease outbreaks in different parts of the world to a specific food source. FoodNet, active surveillance network used to follow 9 major foodborne diseases. It enables public health officials to rapidly trace the course and case and cause of infection in days rather than weeks. Fermented foods. Alcoholic beverages. Alcohol is produced from fermentation by the yeast Saccharomyces cerevisiae, and there is bread, dairy products and other fermented products. Beer. Beer is beer. It is produced by the fermentation of malted grain. Now what is malted grain? It is a grain that has been allowed to germinate, then dried in a kiln and perhaps roasted. Germinating the grain causes the production of a number of enzymes, most notably alpha and beta amylase. Malted grains that may be used as barley, rye or wheat. Unmalted grains such as rice or corn may also be used. The grain is ground into a grist and mixed with heated water in a process called mashing. A series of temperature changes, mash rests, activates different enzymes that in return change the mash to produce desirable characteristics as well as fermentable sugars. Mash rests 49 to 55 degrees Celsius, generally 120 to 130 degree Fahrenheit. It activates various proteinases. Too much protein can make the beer hazy, but some protein has to remain to produce a head on the beer. 60 degrees Celsius to 140 degree Fahrenheit activates beta gluconase, which breaks down gummy beta glucans and allows sugar to flow more freely from the grain. Fungal beta gluconase may be added as a supplement. 65 to 71 degrees Celsius, that is 149 to 160 degree Fahrenheit, it activates amylases that convert starch into fermentable sugars such as maltose. After mashing, the spent grain is separated from the liquid. The grain is usually sold for livestock feed. The liquid at this point, called wort, is transferred to a large kettle where it is boiled with hops and perhaps other herbs or flavors. After boiling, the wort is clarified by spinning it in a whirlpool like a continuous flow centrifuge and transferred to a fermentation tank. In the fermentation tank, yeast is added, which is the process known as peached. Top fermenting yeast produces ales. Bottom fermenting yeast produces lagers. After fermentation for 1 to 3 weeks, the green beer is transferred to conditioning tanks where the yeast and other particulates are allowed to settle and the beer is carbonated. The next is wine. Wine is fine, produced from the fermentation of fruit juices, usually from grapes. The grapes are crushed to form a must. For white wines, white grapes are usually used and the skins are removed from the must before fermentation. For red wines, red or black grapes are used and the skin is allowed to remain during fermentation. For rose wines, red grapes are used and the juice is allowed to remain in contact with the skins just long enough for a rose or pink color to develop. The must undergoes primary fermentation. Natural yeast on the skins of the grapes may be used, but in commercial production, cultured yeast is often used to give more predictable results. The amount of sugar in the must during fermentation is measured with a saccharometer, a calibrated hydrometer. Malolactic fermentation by bacteria in the must converts malic acid into lactic acid. After primary fermentation, the mast is pressed, red wines, and transferred to different containers for secondary fermentation. Secondary fermentation and aging. It takes 3 to 6 months, done in either stainless steel vessels or in oaken barrels. The vessel is kept airtight to prevent oxidation. Proteins are broken down and particles settle. Blending and then bottling. Distilled spirits. Liquor is thicker, produced by the fermentation of grain mash, similar to beer, followed by distillation to increase the alcohol content. Different types of grains are used to produce different types of whiskey. Bread. It involves growth of Saccharomyces cerevisiae, Becker's yeast, under aerobic conditions. It maximizes carbon dioxide production, which leavens the bread. Other microbes used to make special breads, example soda bread. It can be spoiled by Bacillus species that produces ropiness. Yogurt. Milk is fermented by a mixture of Streptococcus salivarius species, Thermophilus and Lactobacillus bulgaricus, official name Lactobacillus delbrueckii, subspecies bulgaricus. Often these two are co-cultured with other lactic acid culture bacteria for taste or health effects. Probiotics. These include L. acidophilus, L. casei and Bifidobacterium species. Acid produced from the fermentation causes the protein in the milk casein to coagulate into a semi-solid curd. If you want strawberries or peaches, you must add them after the yogurt is made. Cheese. Milk is treated with lactic acid bacteria and an enzyme called rennin that partially hydrolyzes the protein and causes it to coagulate into curds. Liquid portion of the milk at this time is called whey. The whey is separated from the curds and the curds are aged, that is ripened. Different microbes in the early and late stages of processing give rise to cheeses with different characteristics. Other fermented foods include sausages, hams, bologna, salami, ijushi, fish, rice and vegetables, katsuobushi, which includes tuna, and sauerkraut. Thank you. With this, I am ending the session. If you like the video, please hit the like button and subscribe the channel and please share with your friends. Once again, thank you very much.